what I would like to do in the next 20 minutes is to address the question of whether we can identify signatures, and everybody talks about signatures, by the use of metabolite analysis, which potentially explain better the function of this complex topic of the microbiome. I want to remind everybody, and I know that Hani gave this morning already an introduction. I'd like just to remind us, microbiome research is now 20 years old. It started more or less with this definition here on what the microbiome really is in a modern sense by Joshua Lederberg and Alexa McRae. And then it started off, plenty of publications. A lot is correlated with changes in this microbial ecosystem, especially in the intestine, and it's linked to metabolism, nutrition, that's, I think it's understandable, but also inflammation-related diseases, infections, even gut-brain axis is affected. So the question is, can we explain changes in the sense of biological functions? And I think that's a huge challenge. The whole modern way of looking at the microbiome is 20 years old, and it's really not enough to explain all these correlations, and we're just at the beginning. And to illustrate this, I would say what the host genome research did 20 years back, where genome-wide association studies started, the microbiome field is right now. You see here two larger population studies, a Chinese cohort, a cohort which we had in hand. It was already introduced in the region of Augsburg. And if you look at here 7,000 people, and here you look at 2,500 people. And at the moment, you just do this at the level of a phylum resolution of the bacterial taxonomy. And phylum is really galaxy-wide views into this. And at the end, you should come to strain level in order to understand the functions. But what you can see, even at the phylum level, you have various different ranges of major phyla in their ratios, firmicutes to bacteroidetes. And that already indicates how variable the whole individual microbiome is. And people tried now in cohort studies to identify features which explain this variation. And you can see here a whole bunch of it. And these features explain 15% of the variation in the Chinese cohort. And if you look at this bar here, regional differences contribute massively here, 6 and more % to the total variation. When we look into the cohort here, we can explain only 9% of the variation, but we had a regionally confined cohort. And again, physiology, disease, lifestyle, medication all contributes to the variation of the individuals. And still, we don't understand more than 90%. And that, I think, is a scary number, because a lot of correlative analysis and suggestions are around. To give you another sense of how we look at these things, this is a phylogenetic tree here, and all these samples were run on our own platform. So there is no methodological variation introduced. This is a meta-analysis of now 4,500 human individuals. And if you look at all these samples and you follow here these lines, and then you have to see that the farther away these patients really are when you follow the lines, the more distant are their phylogenetic composition and the more distant are their microbiomes. But what I want to say is, if you then plot diseases around here in the outer circle, and we compare the inflammatory bowel disease to metabolic diseases, you would expect that there is a clear cluster, yeah? Because if you would look into Crohn's disease or ulcerative colitis, severely inflamed patients, you would expect that something at least would cluster. And at the end of the day, what you see is that the patients basically cover more or less homogeneously the whole phylogenetic diversity of this population. And help me out, if you see a pattern, I don't see one. And that's one of those conclusions, that individual variation in these sequencing-based analysis actually masks a lot of putative signals. And you see here another measure of how we look at microbial profiles. We just count, for example, the taxa we find, that's called richness. And again, what you see here is this massive variation of healthy, of obese, of metabolically challenge people of people with inflammation. And if you plot suggested ratios of bacteria, it doesn't really look better. You have massive variation between or within these cohorts of people. So the question is, can we extract information from the taxonomic analysis based on sequencing? And this is a funny illustration, I find, but what it should show is that you can share a lot of genetic similarities. These two fellows share 98% and more in their genomes, and phenotypically they're very different. These two fellows, which are 99% similar, share very little genomic information with this fellow here. Still, phenotypically, they behave and look the same. So the point is, can we really extract information from only sequencing? And I guess you already see my bias. I don't think that we can do this. And the reason is that the differences and the variations are just not allowing clear separation of marks and features. So what I would want to give you is a little example on inflammatory bowel disease. And I think this is a paradigm of modern diseases because it's complicated. There's a genetic component to it, but nevertheless, the environment is a very important driver of disease onset and progression. But the biggest problem is that these types of diseases are not continuous. They have flares of active and inactive disease. They remodel organs in which the disease is manifesting. So you have an involvement of a certain disease. So can you compare from one individual, if you take a sample here, or if you would take a sample here, you probably would find completely different signatures. And what you see here is a cohort of Crohn's disease patients which have been severely ill. They underwent an allogeneic stem cell transplantation. And the reason why we chose that is you really silence the disease, and then you can look in a follow-up and compare, again, disease progression. This is a 16S profiling pattern of the samples which we had. We followed one patient which started here with severely inflamed conditions, then went in this treatment scenario. The disease went into remission, and then we followed the microbial profile, and you see how dramatically these samples along a 5-year time course change. Here the patient relapsed, received medication. Again, you change again, complete the profile. So this one patient went through the whole phylogenetic diversity of the whole population of patients illustrated here. What do you do with this kind of information? Are you able to identify signatures which predict response to therapy or the risk to relapse or the progressive phenotype? And that's very difficult. And if you only look at 16S sequencing, this is a 16S profile where you plot red the active disease patients and blue the inactive situations against each other. And you see that these ellipses separate okay. You do a differential abundance analysis. You pick certain taxa which are associated with active or inactive disease. And then you use machine learning. That's a random forest classifier, which tries to predict activity of the disease. And if you look at the curves and the areas under the curve, then you receive numbers around 82% area under the curve. And that's okay for populations. That's far not good enough to have an individual signature predicting a disease course. We went on and used untargeted metabolite profiling to see if we can add information into this system. And here is a PLSDA analysis of metabolites of patients and their samples. You would say, yeah, you can separate. This volcano plot again shows you metabolites which are differentially abundant in active or inactive. And one feature came out, and that's the sulfur-containing metabolites, which obviously seem to be enriched here in this active group of samples. Correlation analysis, and that's one take-home message. You always need bioinformaticians in this modern game of analysis. In this correlation analysis, we identified 2 clusters. Again, red enriched in patients with active disease, and blue clusters in patients with inactive disease. And these bacterial metabolite data integration targeted a number of different bacterial species or families. One spotted out here, the Vibrio, and I come back to this in a little second. When again we used random forest classifying tools, you see that the predictive value using this additional information really improved the classification of disease activity. So you can improve your predictive value, including different information, but the functionality and the translation, I think, is equally important. Here we have to jump into models. And this is why we are so terribly slower than all the people in the bioinformatic fields, because they have now access to quite a wealth of data sets, and they run data analysis, and then we try to make sense out of this in models. And one is the use of germ-free rodent models, and this is something which should illustrate you that this type of research is not only 20 years old, it's 100 years old, more or less. And this is Life magazine in the 50s, illustrating the first germ-free rats, which have been made germ-free in a way which we at the moment cannot even understand anymore, in very sophisticated but large chambers here, where you had to dive through decontamination solutions to go into chambers and then to work with these animals. But one take-home message: these animals can live without microbes. Nevertheless, microbes influence us in a tremendous way. So long story short, we use germ-free mouse models. And I go back to this population of patients, and we use samples, again, active and inactive, and put them into disease-susceptible animals. And one take-home message here is that even if you transfer complex communities of bacteria, they are functionally very different. If you transfer samples from active disease patients, or if you transfer samples from inactive patients, then you see that the disease activity in the recipient mice is very different. And I could give a whole lecture on how sensible or not sensible these types of mouse models are, and I think they are very informative. But nevertheless, they are discriminative in the way that they reproduce disease activity from patients. And from this information on, you can work yourself through 16S sequencing on the level of microbiota profiling. You can use untargeted metabolomics, which is, again, when you look at the color codes, the untargeted metabolomics gives you clearer clusters of situations. And this is, for example, active disease, this is inactive disease, and these are conditions still active, but after treatment. And if you look at the different clusters which we get at the 16S level, it's more diffuse, so you can gather more consistent information by integrating metabolomic information. And we then look at these integrated data sets. We were able to identify in the mice certain taxa which seem to be enriched in an aggressive situation. And the aggressive situation is the inflamed situation. And similar to the humans, we identified the Vibrio as a genus which seemed to be strongly enriched in these mice, having then an inflammatory phenotype. The trouble starts now, because if we look at the genus level, still under that umbrella, we find, when we look back into the patient samples, we not only find one genus of the Vibrio, we find a whole set of different genera, and this is now metagenomic data sets. So we're trying to extract even more information from the genetic side, digging further down in resolution, and if you're using these metagenomic data sets, you can at least specify species, if you're a little lucky, even strains. But even here at the species level, you see that there is not only one Vibrio in these patients, they're very different. The patients have different combinations of Vibrios. And for people who work with bacteria, strains matter, and strains really affect at the end functions. So the pledge here is that we are now at the level of resolution where we finally enter again from humans to mice, that we enter a strain level, hopefully resolution, because we identify now on isolate single strains on agar plates, which is tedious work. And it takes us more than a year to extract pure Vibrio strains from the different patients. And at the end of the day, I would like to give you in the last slide one further aspect which we need to consider when we talk about diseases, because the bacteria have a spatial resolution in the intestines, in humans and the mice. And if you look here in a mouse intestine, for example, and here we compartmentalize the different parts of the colon here, and you see in color codes here the different abundances of Vibrio. And here we defined a Vibrio which we actually don't know what it is. Here it's at least a species which we have specified, but in both situations you see that in the proximal part of the colon there is a higher level of abundance of these Vibrio compared to the distal part. So there's a clear spatial resolution which is then likely linked to a certain functional aspect. So this is looking back 5-6 years of work based on first data coming from profiling technologies. And my pledge here is that these, both on the metabolite profiling level and the 16S profiling level, they give very good information, but they are in most cases correlative. And then you have to clearly define patient subsets, which you take samples and take them all the way through these profiling technologies and then transfer them into model situations. And the models have their downsides, but I think they are absolutely required to understand at the end a strain level of functionality, which we then can translate back into the patients. And looking at the progress we have made in this field, also in inflammatory bowel disease, after 20 years of genomic research, I think the message is very clear. There's not a one size fits all. It's very patient individual. It's even within patients a longer time course variation, which needs to be considered if you want to make sensible contributions of how bacteria influence the disease. So having said that, I think there is still a long way to go. I think the microbiome research has consolidated in a way. I think the correlation hype is still visible. You can still feel it, but people understand that correlation is not everything. And the hard work starts in many ways now. We have learned this from the human genomic research that just looking at SNPs is not explaining much at the end. It just gives you indications. So having said that, thanks a lot and happy to take questions.